Okay, so welcome back to this next video in which we are discussing gastric acid secretion. Okay, so we've discussed that controlling the parietal cell's secretion of hydrochloric acid is the second type of cell known as an enterochromaffin-like cell or an ECL cell. Okay, and the way that the enterochromaffin-like cells control the release of hydrochloric acid by the parietal cells is through the secretion of histamine. Okay, so the amount of hydrochloric acid which the parietal cells secrete is directly proportional to the amount of histamine that you bathe them in, basically. Okay, and we now want to see how does stimulating parietal cells with histamine trigger them to secrete hydrochloric acid. Okay, so we've discussed that histamine acts on H2 receptors on the surface of the parietal cells, and what then happens is the H2 receptors undergo a conformational change, and the intracellular loops, all three of them, now become capable of interacting with the heterotrimeric G protein, and the specific heterotrimeric G protein which they interact with is a GS heterotrimeric G protein, which only tells us what the alpha subunit is. It tells us that the alpha subunit is an alpha S subunit. Now what happens is initially the inactive heterotrimeric G protein, in which the alpha S subunit has GDP bound with the beta and the gamma complex, now comes and binds to this special binding domain on the cytoplasmic side of the H2 receptor. And when it binds there, what happens is the alpha S subunit leaves the GDP, okay, so the GDP is released from the alpha S subunit, and instead a molecule of GTP comes in from the cytoplasm and binds there instead. And now once the alpha S subunit has GTP bound, the intracellular side of the H2 subunit no longer wants to bind to the heterotrimeric G protein, and furthermore the alpha S subunit no longer wants to bind to the beta gamma complex. So what happens is the alpha S with the GTP bound goes off, it leaves the H2 receptor, and so does the beta gamma complex. And now both of them will have downstream targets, but the important one as far as we're concerned is the alpha S GTP subunit. This is going to go and interact with enzymes which are in the membrane of the parietal cells, known as adenylyl cyclases. Okay, so it's going to go and activate adenylyl cyclase enzymes. Okay, now let me discuss with you the structure of an adenylyl cyclase enzyme. So basically, adenylyl cyclase enzymes sit within the lipid bilayer, okay? So let me draw one out for you. So here is the amino terminus, let's say, of the adenylyl cyclase polypeptide, and then this line will represent the polymer of amino acids. Okay, so it crosses the membrane once, and it's going from the cytoplasmic side to the extracellular side, and then there are 6 more crossings, like so. Okay, then you have a special little loop here known as the C1 domain, which is on the cytoplasmic side, and then 6 more membrane-spanning alpha helices, all clustered again together, like so. And then you have another special domain which is on the intracellular side before you then finish with the carboxylic acid terminus. Okay, so basically this loop between these 2 clusters of 6 membrane-spanning alpha helices is known as the C1 domain. Okay, this tail that's on the cytoplasmic side down here is known as the C2 domain. These 2 clusters of 6 membrane-spanning alpha helices, they also have names. This is known as the transmembrane domain 1. Okay, so all of these membrane-spanning alpha helices are all clustered together under this name, transmembrane domain 1. Okay, and again, all of these 6 membrane-spanning alpha helices over here, which are all clustered together, are called the transmembrane domain 2. So you have the amino terminus, then transmembrane domain 1, then the C1 domain, then transmembrane domain 2, and then the carboxylic acid tail here. Now, C1 is split into 2 separate domains. Okay, so you can split C1 further into C1a and then also C1b here. And similarly, C2 can be split into 2 separate sub-portions, okay? So you can split it into C2A here, and then also C2B. So both C1 and C2 have 2 portions. Now basically, the active adenylyl cyclase enzyme is made from the dimerization of the C1A domain with the C2A domain. Okay, now what do I mean by the active adenylyl cyclase enzyme? Well, basically, adenylyl cyclase enzymes catalyze a certain conversion. Okay, so they take ATP molecules, and let me just draw you a little cartoon of ATP molecules. So ATP stands for adenosine triphosphate. So the A is for adenosine, and then the TP is for triphosphate. Okay, now adenosine means ribose bound to adenine. Okay, so it's the name for ribose, which is this pentameric ring with the 5th carbon coming off up here, then bound to the organic base adenine. Okay, so this is what adenosine is, adenine with the sugar ribose. Then triphosphate means that we've got 3 phosphate groups coming off this 5th carbon's alcohol group over here. Okay, like so. So let me colour in the different portions here. So these are the 3 phosphate groups here, 1, 2, 3, and they're labelled the alpha, the beta, and the gamma phosphate. So the first phosphate is called the alpha, the second is called the beta phosphate, and the third is called the gamma phosphate. Okay, now I'll colour in the ribose sugar in blue here. Okay, and make note that that is ribose, not deoxyribose. It's the sugar that is used in RNA, not the sugar that is used in DNA. Okay, and then you finally have the adenine organic base over there, which I'll colour in green. Okay, now all of that together is adenosine triphosphate. Basically, what adenylyl cyclase enzymes catalyze is they catalyze the conversion of adenosine triphosphate into adenosine monophosphate, but the cyclic version of it, so cyclic adenosine monophosphate. And for short, cyclic adenosine monophosphate is abbreviated to CAMP, or cyclic AMP. So this is cyclic adenosine monophosphate. Okay, so if we were going to make just adenosine monophosphate, then that we know instantly how to draw that structure. We know what the structure of adenosine is, and monophosphate just means that it's only got 1 phosphate group coming off, so that would just be the adenosine structure with a single phosphate group coming off. However, we want cyclic adenosine monophosphate. Now that makes it slightly more complicated, but what we're going to do basically is if I draw the ribose sugar here again, okay, so here's the 5-membered ring, here's the 5th carbon coming off up here, here's the organic base adenine. We're basically going to have a phosphate group that connects the 5th carbon here to the 3rd carbon down here, like so. Okay, so here's the ribose in blue, like so. And then we have adenine, the organic base adenine in green here. Okay, and then we have a single phosphate group between the 5th and it's the 3rd carbon of ribose down there. Okay, so this then is cyclic adenosine monophosphate. It's adenosine monophosphate because we've got adenosine here, ribose plus adenine. Okay, so I never managed to complete this. So ribose plus adenine here makes adenosine. Okay, and it's monophosphate because we've got a single phosphate group, but because we've got the single phosphate group bound again to the alcohol group that comes off the 3rd carbon of the ribose sugar, it's then cyclic adenosine monophosphate, or cyclic AMP, or CAMP. Now, of course, when you do this conversion, you're also going to break off the beta and the gamma phosphate. They remain bound together, like so. And when you have 2 phosphate groups bound together, this is called pyrophosphate. Okay, so this is a pyrophosphate molecule. So basically, adenylyl cyclase enzymes catalyze the conversion of ATP into cyclic AMP and pyrophosphate. Okay, right. the active enzyme that catalyzes this is made from the dimerization of this C1a domain and this C2a domain. Okay, so the TMD1 and the TMD2, although they look mightily impressive sitting here, they are just to attach the enzyme into the lipid bilayer. They're not actually the portions which catalyze this reaction. The portion that catalyzes this reaction is made from the C1a and the C2a domains. When they come together, that will then make an active enzyme which is capable of catalyzing this reaction and hence producing cyclic AMP. Now, before we go any further, I'd just like to say that there are 9 known forms of adenylyl cyclase called adenylyl cyclase 1 all the way down to adenylyl cyclase 9. So it's very, very simple nomenclature for the adenylyl cyclases. And all of them are activated by alpha S GTP subunits. So how does the alpha S GTP subunit activate the adenylyl cyclase enzymes? Well, basically, it will come over and it will bind to both the C1 domain of the adenylyl cyclase and it will also bind to the C2 domain, and basically by doing this it forces the two to come close together, and then what happens is the C1a will then dimerize with the C2a. So basically it binds to both the C1 and the C2 domains, keeping them, which forces them to come nice and close together. And then what will happen is C1a will dimerize with C2a, and then that sets up an active enzyme which is capable of churning out cyclic AMP. Okay, so let's just draw the update basically. What will happen is, if this is the plasma membrane again, what will happen is the C1a domain will end up very close to the C2a domain, and this is because the alpha S GTP subunit has bound to both the C1 domain and the C2 domain. Okay, so here's transmembrane domain 2, and now what I want to try and show is C1a and C2a close together, like so. This is the best I can do really, without being able to draw in 3D. Okay, so here are C1a and C2a, which are associated together, and here is the amino terminus, and here is the carboxylic acid terminus. Okay, so I'll just label up the portions again. So this is transmembrane domain 1, or TMD1, and this is transmembrane domain 2, or TMD2. This here is C2a, and this here is C1a. Okay, and they've dimerized together, and once you have this dimerization, this will form an active enzyme which will start churning out cyclic AMP. Now, what that then leads to is a huge production of cyclic AMP within the parietal cells, and what we now need to look at is how does that cyclic AMP then trigger the activation of hydrochloric acid secretion by the parietal cells. Okay, so basically it does it by activating protein kinase A enzyme. So this is the target for it's one of the main targets for cyclic AMP, protein kinase A. Now basically there are 2 different types of protein kinase A. There is type 1 protein kinase A, which is free within the cytoplasm, and there is also type 2 protein kinase A, which is instead bound to special proteins which are bound to the plasma membrane, known as A kinase anchoring proteins. Okay, so if this is the plasma membrane, there are special proteins here, okay, which are bound to the plasma membrane, known as A kinase anchoring proteins. And for short, A kinase anchoring proteins are often abbreviated to ACAPs. Okay, so A kinase anchoring proteins, or for short, this can be called ACAPs. Okay, now there are a huge number of different ACAPs, but the important thing to understand about them is they bind type 2 protein kinase A. So what I want to now show is the 2 different types of protein kinase A. So I'll start with type 2 protein kinase A, this one which will be bound to the ACAPs and is therefore sequestered at the plasma membrane. Okay, so protein kinase A is not just one little protein. It's a huge, well, it's a complex of 4 different proteins, basically. Now, what the important thing to understand is that this complex of 4 proteins is actually the inactive form of protein kinase A. What's going to happen is 2 of these subunits are going to dissociate away from the other 2, basically, and those 2 which dissociate away, they will be the active portion of the protein kinase A, which actually catalyzes the addition of phosphate groups onto serine and threonine residues of proteins. Okay, so let's just try and put a little bit of colour onto this. So in turquoise here, we have the A kinase anchoring protein. Okay, and here I am showing a type 2 protein kinase A molecule. And basically type 2 protein kinase A consists, well, all protein kinase A's consist of 4 separate proteins. We've got one here in this L shape, another identical one here in this L shape, and then 2 here which I've shown as balls. Okay, now these 2 that are in the L shape, these are known as the regulatory domains, okay? And basically, because we are talking about type 2 protein kinase A, which is attached to the A kinase anchoring proteins, the regulatory domain that you will have for type 2 protein kinase A is called the R2 domain. Forget domains, it should be subunits rather than domains. Okay, so these are the regulatory subunits. So we have 2 R2 regulatory subunits. Okay, so let's colour those in. So, in red here is one of these R2 regulatory subunits. Okay, and then we have another one over here, like so. Right, okay, so if you want to make a type 2 protein kinase A molecule, you need 2 of these type 2 regulatory subunits of protein kinase A, and then you also need 2 of these catalytic subunits, which are these balls in here. Now, the important thing to understand about the difference between protein kinase A1 and protein kinase A2 is that the difference is purely in the regulatory subunits rather than in the catalytic subunits. So the actual portion which adds phosphate groups onto serine and threonine residues within proteins is identical between the 2 different types of protein kinase A. Okay, and we'll call it there for this video and continue our discussion in the next video.